So I'm interested in how the central nervous system, in particular the brain, develops, and how you can get this immense diversity of cell types that we have in the brain, with hundreds, if not thousands, of different cells. Stem cell is sort of like the building material of your whole body. And what's typical for a stem cell is that it first of all has the potential of dividing and expanding and making more stem cells at a very high rate. And the second thing is that it's multipotent, so a stem cell can become a nerve cell, or it can become a muscle cell, or a liver cell. So it has that multipotency, and it has the eternal life of being able to replicate. Looking into elderly population, when your brain starts to unfortunately degenerate to some extent, and there's various types of problems such as Alzheimer's and other things, where bits and pieces of your nervous system are essentially falling apart and falling away and dying. And we're hoping that by understanding how they're generated from the beginning, that maybe we can make new such spare parts to put into the brain in a petri dish, and hopefully in the future even replace those damaged cells in elderly patients and for various types of diseases. That is already in clinical trials, so there's a way nowadays of taking either blood cells from the patient or biopsies of the skin. And those cells, which will be blood cells or skin cells, they can be reprogrammed back to stem cells, so they program back in time to a more primordial type of identity, and then driven along different sort of avenues into different, for instance, nerve cells. A fair number of children are born with various types of developmental issues with the central nervous system, and we're very interested in trying to understand what underlies those problems and why certain children are born with some developmental problems with the brain, and trying to understand what genes are underpinning those problems. And so we're in a very interesting era now, moving forward the next 5-10 years. And our work understanding sort of gene by gene will help to provide information about the role of every gene, so that when you see such a gene affected in a human being, you'll know what it normally does. And so we're sort of still in this big mapping adventure right now. We're sort of understanding what genes do, and then mapping which ones of them are affected in various patients. And I think it's something that more and more people are looking into doing, and I think it's something that has a lot of potential.